Hello and welcome to Quick Anatomy Hub. We have discussed fertilization in previous video and we have seen the formation of zygote there. Moving forward from there, we will discuss the division in zygote, cleavage, blastulation or blastogenesis and implantation in this video. Almost after 30 hours of fertilization, series of mitotic division occurs in zygote. The cells there are called blastomeres, which are retained within the zona pellucida. The peristalsis in fallopian tubes continues to move this dividing ball of cells to the uterine cavity. On third day of fertilization, there are 12 to 32 blastomere clustered together, that is called as morula. On fourth day of fertilization, morula reaches uterine cavity. It starts accumulating fluid from uterine cavity through fenestrations and zona pellucida. This will result in the bunch of cells that will get compacted to one side of the ball and the group of cells lining the edge of this ball inside the zona pellucida. This process is called blastulation or blastogenesis, and the resulting product formed is called blastocyst. Here you can see the compacted cell mass to one side. It is called inner cell mass or the embryoblast, and the cells outlining this ball, it is called the outer cell mass or the trophoblast. As the blastocyst floats in uterine cavity, there is degradation of zona pellucida, facilitating the rapid growth of blastocyst. The embryo absorbs nutrient from the uterine secretions now. On day 6, the trophoblast differentiates into cytotrophoblast and syncytiotrophoblast. Here you can see the cells from the trophoblast starts to proliferate and they extend their papillary projections into endometrium, releasing an enzyme that breaks down the maternal tissue for further implantation of blastocyst. This part is called syncytiotrophoblast. Now this will make connection with the maternal blood vessels and gets their nutrition from there. The part of the trophoblast outlining the embryoblast is called as cytotrophoblast, and this is how implantation occurs. Here's a quick review. We have seen mitotic division in zygote and formation of morula, which enters the uterine cavity, and then blastogenesis occurs. The formation of outer and inner cell masses, the degradation of zona pellucida, and then the trophoblast, which divides uh, into cyto and syncytio trophoblast, which will then invade the uterine wall to get implanted there. The inner cell mass or the embryoblast will then form the bilaminar disc that we will discuss in next video. Hopefully this will be helpful for you. If you have any question, you can comment below. Thank you.